Can you guys hear and see me?
Yep.
Hi, good morning.
Morning.
Let's see here. Okay. I want to say something really quickly here. I was asked last week during our lab how much of what we were studying did we, with regards to terminology, did we need to learn? And I thought about that over the weekend. And the more that you can learn right now, the easier this semester will be and next semester will be for you. And the reason I say that is because, for example, in lecture, we're going to spend a lot of time learning the physiology. But in lab, we're going to focus mainly on the anatomy, the majority of the time on the anatomy. And if you can learn the terms that were covered in this Lab 1 PowerPoint, and I'll show that to you again here. Give me one moment here. The more of it you can learn right now, the easier your, when we start talking about identifying the muscles in lab and we identify the bones of the skeleton in lab, the more of this that you can learn right now, the easier that will be. And the reason I say that is because a lot of muscles and bones are named according to where they are, using this terminology. For example, one of your biggest muscles in your butt is your gluteus maximus. And then meaning maximus, meaning a big muscle right here. Your gluteus minimus is the little muscle that's right here. If during an exam you put down part of that, put down either gluteus or maximus or minimus there, you get partial credit. Anatomy is a word puzzle. If you can learn these regions, you can often be able to identify what's also in that region because it's named similarly. From the veins to the bones to the muscles to the nerves. They're named very similarly. Another example is this little area right here, back of your elbow. This is your olecranium. On your ulna here, if you grab the back of your elbow right here, grab your elbow, and you know that in your arm here you have a radius and an ulna. When you flip over your arm, when you're in a bear with me, you're in a supinate position where you're holding a bowl of soup in your hands. You pronate your arm. So supinate, pronate your arm. Which bone do you feel move? Your radius is right here, your ulna is right here. Which bone do you feel move, and which bone do you feel that doesn't move?
The ulna.
Which one moves?
The ulna moves.
The
radius. The ulna stayed the same. The radius moved.
Exactly. And that part on the back of your arm that you're actually holding, this little, where your elbow is here, this little ridge, that's called the olecranian process. And if you look right here on the PowerPoint, that area is called the olecranian. Thinking back on going back to your radius and your ulna here, your ulnar, if you know that this is the radius, the ulnar artery runs right along there. The radial artery runs right along your radius. You will learn that a lot of your anatomy and your bones, your nerves, your blood vessels, your muscles are named according to where they're at. But they do use this terminology to name them. What I would do if I were you to learn this terminology is I would print out this sheet. And I would look at it 3 times before you go to bed. Go through and name them off. Look at them, point them out to yourself, and name them off. And when I say name them off, I'm saying speak out loud. Say the name. Do it 3 times right before you go to bed. And do it once the next morning. And I can almost 99% assure you that within a matter of 3 or 4 days, you're going to know all these terms. And the reason I do that is because if you ever thought about right before you go to bed, during the night, what do you think about? You think about what you did right before you went to bed. And in your subconscious, you will replay this material over and over again. And then throughout the semester, as we learn about bones here in about 2 weeks, and we start going through naming the bones, that will come a lot easier to you if you know this material right here. I'm not saying to memorize it all at this very moment, but I'm saying the more that you can learn right now, the easier it will be.
I did have a question, Professor.
Go ahead.
I was curious, should we have a notebook for our lab and for lecture, like 2 separate?
I don't think it really matters. Since we're doing remote right now, I don't think it really matters. I would make sure that you keep your PowerPoints for the two separate if you decide you're going to print them out, so that when it comes to studying, it makes your life a little easier, because you're going to have lab exams and lab quizzes, and then you'll have lecture exams and lecture quizzes. If I hit on the same things in lab that I touch on in lecture, you can almost be assured that's going to be on the exams and quizzes. Because if I'm going to try really hard to tie in what we study in lab with what we study in lecture. And when I'm going over that material more than once, I'm going over it in lab and then I'm going over it in lecture, you can almost be assured it's going to be on an exam or a quiz or something. How many of you, I know we struggled to watch the videos before, and I realized once we were done here that I did not put them on there right. I tried to copy them the way that they had been copied on there before by another professor, and it didn't work. But over the weekend, I went through and put them all up there correctly. Is there anybody who can't play the videos on their own at this point?
It's okay. I hear the game.
Okay. Let's start into is there any more questions about lab right now, or questions even about lecture?
I have a question.
Go ahead.
So for the recorded Zoom meetings, I went to them on the content section where it says Zoom recorded meetings, but there's nothing there.
Not on there yet. Be patient with me. I will work to get them up this weekend.
Okay. Okay.
A lot of this is new to me, learning how to do D2L, learning how to record Zoom meetings and learning how to attach them is all very new to me. I was shown this on, I think, Thursday night of last week, how to do it. I just need to get it done. Bear with me as I get it done, and I will get them up there.
Okay.
Now, get out of this. Sorry, just figuring out how to get out of this one. I think I can do it just like this. Oh, and if you, last night, or I think it was the night before, I got up the worksheet for chapter 2. Yes, it's a lot of terminology, it's a lot of vocabulary. We will start working on that again on next Tuesday. And I realized after I ended our class on Tuesday that I let you out early, and we should have gone until 10:45. And I think we ended around 10:15. So I'm not going to do that again, just to keep us on schedule. So the instructions for this is to watch the videos, then log in to Connect and complete the Lab 2 activities. Download the 3 worksheets for Lab 2, and you'll be completing them with your lab partners during breakout sessions. I haven't assigned the breakout sessions, but we can do that today. So this first video is about the compound light microscope, and for some of you, but it was a while ago that you took biology. For some of you, it was maybe last semester. Let's, if you have problems, just let me know. But what I honestly I need you to do is I need you to review your past biology textbooks. Any material that you have that within the next 2-3 weeks. A lot of this is a review of Biology 101 and Biology 106. Oh, why do we not have any sound? There we go.
This is a compound light microscope. We use a compound light microscope to look at structures that are really small, like cells or tissues. The term compound refers to the fact that it uses two sets of lenses. There's the first set of lenses, which are called the ocular lenses or the eyepieces. This is where you're going to actually put your eyes in order to view the image. And there is a second set of lenses called the objective lenses. So both lenses work simultaneously to magnify the object. The term light refers to the fact that it uses a beam of light that shines up through the specimen in order to visualize the specimen. That's why the specimen or the tissues, the cells that you're looking at have to be very, very thin in order for the light to pass through. If the specimen or the slide is too thick, not allowing light to pass through, then you won't be able to see anything. But here you see we have a thin section of tissue that's on this microscope slide. There are three main parts of the microscope. There is this foundational support called the base. There is this curved portion called the arm. And then there is this final portion attached to the arm called the head. What we find on the head of the microscope are the ocular lenses, the body tube, and this metal turret that revolves, and this is called the revolving nose piece. And again, it contains those 4 objective lenses that are mounted onto it. We turn the microscope on and off by this switch at the base. Also on the base is a dial, and you can see that this dial regulates the amount of light coming up through the light source, which is also embedded into the base of the microscope. So here is the light source, and this dimmer switch is called a rheostat. So you can use this to either increase or decrease the amount of light that's coming up through the object. If we turn the microscope this way, we can look at the ocular lenses and the objective lenses. Whenever you use the microscope for the first time, you always want to start with the shortest objective lens over this platform called the stage. This shortest objective lens is indicated by a red band, and it has a number imprinted on it, and that number is 4. So that means that this lens magnifies.
You guys, it's also the shortest lens. All of these, they have lengths depending on their strength. The smallest lens, I've got a microscope up here, 1 second here. This is a really simple microscope. But this microscope right here has three like hers. They usually have three or four. The shortest one is the one that you start with first. It's got the lowest strength, lowest magnifying power.
Magnifies the image 4 times what you would see with your naked eye. This objective lens is called the scanning objective lens. If we turn the revolving nose piece, we see that we have now the next longer objective, and this is indicated by a yellow band. This objective is called the low power objective, and the number imprinted on this objective is 10, which means that this lens magnifies the image tenfold, or 10 times what you would see with your naked eye. The next longer objective is indicated by a blue band. This is called the high dry objective, and the number imprinted on this objective is 40. And that means that when this objective is over the stage, or over the specimen, over the object, that it magnifies the object 40 times what you would see with the naked eye. The longest objective is this objective that's indicated by a white band. And this is called the oil immersion objective. And this is a special objective that we use by putting a drop of oil onto the slide to enhance the resolution of the image. Remember, I told you that this is a compound light microscope. So when we're looking at an image and we want to know the total magnification that we're observing, we need to take into account the magnification of this set of lenses, the ocular lenses, and the magnification of the objective lens, because remember, both lenses are working simultaneously.
So what you do is you multiply the strength of the objective lens and the strength of the ocular lens. So if the strength of the ocular lens is 10 and the strength of the objective lens is 4, you're at a 40x magnification, 40 times magnification, 40 times what your naked eye would see by itself.
To magnify the image. The set of ocular lenses just themselves have a number imprinted on them, and that number is 10x or tenfold. So these lenses magnify the image 10 times what you would see with your naked eye. So if you have the shortest objective lens over the stage, the scanning objective lens, to determine total magnification, what you need to do is take the magnification of the ocular and multiply it by the particular magnification of the objective lens that is over the specimen. In this case, this is the scanning objective, which has imprinted on it 4x or 4 times. And so we would take 10 times 4. And so if you're looking at the image with the scanning objective over the stage, it would be magnified 40 times. 10 times 4 is 40. 40 times what you would see with your naked eye. If you turn the revolving nose piece to the next higher objective, remember that this objective lens is called the low power objective, and it has the number 10 imprinted on it. So if we are observing an image with this objective over the stage, then we have to take 10, that is the magnification of the ocular, times 10, which is the magnification of the objective lens, to get a total magnification of 100 times, or 100-fold what you would see with your naked eye. If we turn the revolving nose piece to the next higher objective, this is called the high dry objective. And remember that this objective has the number 40 imprinted on it. So if we want to determine the total magnification when this objective lens is over the stage, we take the magnification of the ocular, which is 10, and multiply it by 40 to give us a total magnification of 400. So the image would be magnified 400 times what you would see with the naked eye. And then if we move the last objective lens, the oil immersion lens, into place over the stage, remember that the number imprinted on this objective lens is 100. So if we take the magnification of the ocular, which is 10, and multiply by the magnification of the objective lens, which is 100, that would give us a total magnification of 1,000. So this particular microscope can magnify objects approximately 1,000 times what you would see with the naked eye. There are a couple other features that I want to point out. Go back and turn the revolving nose piece so that the shortest objective, the scanning objective, is over the stage. Remember, this stage is the platform on which you are going to put your slide. So here's the microscope slide, and there are clips. These are stage clips that hold the microscope slide into place. So you can just push the slide all the way back, and this stage clip is spring loaded, and so that you can gently just have it go back, and now the slide is securely in place. You always want to position the stained specimen, or the specimen that you're looking at, right over the light. So there is a hole in the stage that allows the light to shine through. And we can adjust the stage by using these stage adjustment knobs. So here you see that we have a top dial, and that moves the stage front to back. And then we have a lower dial that moves the stage left to right. So you always want to position your specimen so that it is directly over the light, and you can do that. The microscope slide is held firmly in place by the stage clips, and then you can move these stage adjustment knobs in order to adjust the position of the specimen so that it is directly under the objective lens. If we look at the underneath the stage, there is what is called a condenser. And the condenser basically converges the beam of light so that it forms a cone of light that comes directly underneath the specimen. And that condenser can be raised or lowered depending on the magnification that you're using. Underneath the condenser is what is called the iris diaphragm. And this works very similarly to the iris of your eye. And so the iris diaphragm has an aperture, this lever, which can be moved to either open or close the aperture to either let in more light or to let in less light. And so we can move that iris diaphragm to either let in more light or less light. And that sits underneath the condenser right above the light source. If we look at the two knobs on the arm of the microscope, there is a large outer knob. This is called the coarse focus knob, or the coarse adjustment knob. And then there is a little inner knob, and this little inner knob is called the fine focus knob, or the fine focus adjustment. Initially, you want to start with your scanning objective over the stage, and you can see that if I move the coarse focus knob, that the stage moves in larger increments. And so you want to bring the image into view by moving the coarse focus knob, this large outer knob. We only use this knob with the scanning objective over the stage, all right? So once you have your image in view and you want to go to the next higher objective, you can move the revolving nose piece into place and then only use the fine adjustment knob. This knob moves the stage in smaller increments, and you can see that as each objective lens increases in magnification, it gets closer to the microscope slide. The distance between the objective lens and the microscope slide is called the working distance. And so you can see that as we increase magnification, our working distance gets less and less. And so with the higher power objectives, the low power objective, the high dry objective, the oil immersion objective, you only want to use the fine focus knob. Otherwise, you risk cracking your microscope slide and then damaging the lenses of your microscope. The other thing that you want to remember to do as you increase magnification is you always want to...
One thing that I would say is anytime that you are no longer using this objective right here, the lowest power objective, unless you are using, let me say it differently, unless you are using this objective, only use the rough focus knob during that, using this objective. Use the fine once you move up from there. Use the fine, so when you change to the next strength and you move up to the next strength of an objective, just use the fine focus. If you realize that you can't find your specimen with the fine focus at that point, go back to the lower objective and use the rough focus again to find it. It will make your life a lot easier if you do it that way.
Use your stage adjustment knobs to center the specimen in the field of view. That's the circle of light that you're observing. As the magnification increases, your field of view gets smaller and smaller. And so you always want to keep the image in sight. And so always make sure before you move to the next higher magnification that you have readjusted your stage, used your stage adjustment knobs to center the image, and then use your fine focus. These compound light microscopes are parfocal, which means that if we initially focus the image using the scanning objective, then whenever we move to a higher objective, that image will stay mostly in focus. All we have to do is fine tune it with the fine adjustment knob.
Is there any questions about that?
This demonstration is showing you the parts of a microscope. We have the eyepiece or ocular. This is a binocular microscope, meaning that there are two eyepieces. These eyepieces can be moved in or out to accommodate the distance between your eyes. We have the arm of the microscope and the base. On the bottom right-hand side is the on-off switch, and on the left side of the base, you're going to find the rheostat or the dimmer switch. This dimmer switch should be rotated so that it's closest to you. And if you needed to dim it, you can rotate it in the opposite direction. We have our revolving nose piece that consists of 4 objectives. The scanning power with a red color-coded band, the low power objective with a yellow color-coded band, the high power objective with a blue color-coded band, and then finally, the oil immersion lens with a white color-coded band. When you're done using the microscope, the scanning power should be left in place. The stage of the microscope, the slide clips that holds the slide. To move the slide, underneath the stage on the right-hand side are 2 knobs that allow you to move the slide back and forth or side to side. Below the stage is the condenser, which consists of a series of lenses as well as a diaphragm that adjusts the light as it goes through the slide. On the base, you'll find the light or the illuminator. To move the stage closer to the objectives, on the arm, you're going to find two knobs. The course adjustment knob that moves the stage in large increments and should only be used for the scanning power. Connected to the course adjustment knob would be the fine adjustment knob that moves the stage in finer increments.
Some of this material can be repetitive from one video to the next, but repetition is frequently how we learn things. And so I would still pay attention even if you think, okay, this was covered in the last video. This next one is using a virtual microscope and showing how things change. It's a video kind of like a movie demoing a microscope that you can log into online and use. Guys, do you have any questions?
That's a lot of different controls to figure out.
It is, and I will tell you that doing this microscope lab virtually is a lot harder than doing it in the lab. And I think you can probably already sense that. It's really about getting your specimen into focus and getting your light so that you can see what you want to see. And being able to, and the higher the magnification, the more you will be able to see. And oil immersion also helps with that. This slide is actually going to show you that what you put under the microscope is actually inverted.
All right, in this video, we are going to be looking at the famous letter E slide. Almost every class that uses microscopes will start with the letter E, just a piece of, from newsprint, wherever. I can give you the details on an activity to do this yourself. But I'm looking at, when I put the slide onto the stage, the E was right side up. It was facing the way that it would look if I was reading it. And here you see it is upside down and backwards. So that's one of the reasons we like to use this letter E, because it shows you that as the light rays are bent or refracted through a microscope, you're actually seeing a mirror image. So just like as I move the stage of the to the right here, you're seeing the letter E move to the left and vice versa. So everything is backwards. That's okay. I'll go into the mechanics of that. But I just wanted you to see this just as a reminder of why we use this activity. So I will go ahead and go to the next power, because the main thing I want to cover in this video is your field of view and how much smaller it gets as we move to higher and higher powers. So this is why it's very important to center whatever you're looking at in the middle of the field of view. So this large circle on the edge, which you can't see, but the entire circle I'm looking at is the field of view. Right now I'm using the scanning power, which is the 4x objective for a total magnification of 40 on this microscope. As I then jump to the low power, as you can see here, I'll get her tweaked a little bit and moved, but you can barely see the entire letter E here. And that's because as I just jumped from the 4x objective with a total magnification of 40 to the 10x low power objective with a magnification of 100, my magnification went up 2.5 times. So my field of view is now 2.5 times smaller. Then as we move forward, and I'm going to go to the high drive power and get to a total magnification of 400x, you're only seeing part of a letter E. And I can fiddle with the light a little bit here, move this around a little bit, but as you can clearly see, you're only seeing part of a letter E, and that's because as I jumped from a total magnification of 100 to a total magnification of 400, so obviously the magnification went up 4 times, and the field of view just dropped 4 times. So that's why we use the letter E, to show us those two things, that the field of view is going to be greatly diminished every time you increase magnification, which is why it's so important to center whatever you're interested in looking at. And number two, the fact that we're actually looking at here is an image that's a mirror image of what we actually put on our stage. All right, I hope that helps.
So whatever you're looking at under the microscope is inverted and turned as what was forward is now backward, and your right becomes your left. So under the microscope, if you move the slide to the left, what you see under the microscope is that it shifts to the right.
In this video, you will learn how to stain eukaryotic cells. To make a wet mount of cheek cells, place a drop of fast blast DNA stain on a microscope slide. Use the blunt end of a toothpick to scrape the inside of your cheeks. Place the toothpick into the drop of stain on the slide and swirl it to release the cells into the stain. Prepare the wet mount by lowering the cover slip over the stain, starting at a 45-degree angle. Avoid trapping bubbles under the cover slip.
So if you put it on the cover slip at an angle, it will push out most of the bubbles underneath the slide.
To make a wet mount of onion cells, place a drop of fast blast DNA stain on a microscope slide. Take a small piece of onion.
I usually tell my students when we do this in Biology 101 or something, I tell you to get the smallest piece you possibly can. Because once you put it under the microscope, it becomes a lot bigger. And you want the light to go through it, like in the first video, she talked about that it needs to be very thin so that the light will go through it. The smallest amount you can will go very far.
Peel a piece off the inside epidermis of the onion leaf scale that is roughly 1/2 to 1 centimeter squared. Place the piece onto the drop of stain.
So I wouldn't even use 1 centimeter squared. I would use almost more like 2 or 3 millimeters. So about 1/3 of what he's using there.
On the slide. Add another drop of fast blast DNA stain on top of the piece of onion. Prepare the wet mount by lowering a cover slip over the stain, starting at a 45-degree angle. If a bubble forms under the slide, additional stain may be added along the side of the cover slip.
So yes, the smallest amount, if it gets too thick, that's when you start not being able to see anything and being able to see what you want to see, based on the fact that your field of view becomes a lot larger under the microscope.
Cells are the smallest living units of an organism. All cells have 3 things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are 2 broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms. such as bacteria. So what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants, that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
Yeah, that will be a really good when we talk about in starting not next week but the following week when we talk about cell structure and the organelles within the cell. That was a good beginning to what we're going to see and what we're going to be looking at and learning about.
It was pretty interesting.
Yeah.
It's kind of crazy how all that's inside us.
Well, and your overall health, because that's the basic unit of the body, the smallest unit of the body, your overall health is always related back to cell function. So when you're looking at your patients here in a couple of years or in a year, their overall health is still leads back to how well are their cells functioning. With me just for a second here. Something over these last videos. Do you remember when we talked about gradients? How do things move? Do they move from a low to a high concentration or a high to a low concentration?
High to a low.
Okay, good. When we're talking about diffusion and osmosis, when there's one exception, when will they move from a low to a high? What do you have to add into the system or the cell?
Energy.
You have to add energy, correct. And so when we talk about diffusion, we talk about osmosis. All this is passive, where things are always just moving from a high concentration to a low concentration based on whatever is diffusing or with osmosis, the water movement. So every time this is just a passive movement of something moving from a high concentration to a low concentration. Oh, really? Wow. This was on here a couple days ago. Let me see here. Okay. Bear with me just for a second here. We're not going to do that video, obviously. I'm gonna do something here just because, and just take it off here. So if you refresh your screens, you shouldn't be able to see that at this point. I think I'm gonna move this one up here. You guys, what is diffusion? How do you define it?
It's like the movement of a substance to a different area or throughout an area.
Mm-hmm. Give me 1 second. I'm going to do a little bit of a demo for you. Give me 2 seconds to prepare it. Right now. I'm going to try to replace that video with a little bit of a demo here. So I have this food coloring that I'm trying not to get all over myself here. And I drop it into some water. You guys see my water? Tell me yes or no.
Yes.
Okay. So I'm going to put just 1 drop in this water here. Maybe. Maybe. Maybe not. I thought there was food coloring in here. Okay. You see where I dropped it in the water? We do 1 more. It's really old food coloring, I think. Okay, you see it now.
Yes.
So I dropped 1 drop in and it was a highly concentrated drop. From that drop, it's going to diffuse out into the rest of the water because it's going to move from the high concentration within the drop to a lower concentration somewhere else within the water. All he's doing every time is just trying to go down a concentration gradient and go from a high to a low. And it's going to continue to diffuse in that water until the concentration throughout all the water is equal and there's no gradients left. So everything in diffusion always moves from a high to a low. When it comes to diffusion, that is the most important thing, is that it's always going to move from a high to a low concentration of whatever substances is. If I put in a yellow drop into this food coloring, it's going to move from a high to a low regardless of the fact that there's blue in there. In if I have a substance dissolved in, if I have a substance, bear with me here, if I have a substance dissolved in water, and I tell you that it is, there's 30% sugar in there. And then I have a cup. I'm going to do one more demo. Give me 1 second here. Okay, so I have 2 containers here. Do you see the water in the containers? Do you see the water in the containers?
Yes.
Yes.
So I have two.
Yes.
I have 2 containers here. One of these containers contains 30% sugar. The other one contains 10% sugar. If they're the same volume, which one contains the most water?
The one that contains 10% sugar.
Correct. If I put them together, and it doesn't work so well with my kitchen Tupperwares here, but if I put them together so that what's in the 10% sugar can move into the 30% sugar, and vice versa, the 30% sugar water, the water can move into the 10%, which way is the water going to move?
From high concentration to low.
So is it gonna, if this is my 10% water, this is my 30% sugar, which has the most water in it. This is my 30% sugar that has less water in it. Where is the water gonna move? Is it gonna move into the 30% sugar, or is it gonna move into the 10% sugar?
10%?
No.
If
80%.
if I have 2 equal volumes here. This one has 10% sugar in it. This one has 30% sugar in it. If this is 10% sugar, this one is 30% sugar, which one contains the most water?
The 30%.
No, because that one contains the most sugar. And if it is the same volume, I'm going to make these a little bit more equal volume. Since I'm doing this right over my keyboard, I'm going to be kind of careful. But if they're equal volume, one of them contains 30% sugar, one of them contains 10% sugar. The more sugar I have in the 30%, the more sugar I have in a volume, the less water I can have in a volume. Is that clear?
Yes.
So if I have 10% sugar and it is able to move into a solution of 30% sugar, it's gonna always move from the higher concentration of water to the lower concentration of water. So osmosis is just the diffusion of water from a higher concentration of substance to a lower concentration.
Okay, so then the 30% sugar would go into the 10% sugar because it has to go from the high to the low.
If we're talking about the movement of the sugar, yes. But what if we're talking about the movement of water?
Okay, then yeah, the 10% then would go into the 30% because that has a higher concentration of water.
Yes. So if we're talking about diffusion, it depends on what our membrane, if we put these together, depending on what is permeable to move through. If it's the sugar, the 30% sugar solution will move in, that sugar will move into the 10% sugar. If it's the water, and we're looking at osmosis, the 10% sugar has more water in it. And so that water is going to move into the more concentrated 30% sugar.
Yeah, that makes sense.
Think through, while we watch these next videos, think through that a little bit, okay? And I'm going to go get rid of my water and my liquids that are right around my keyboard, just waiting to spill over onto my keyboard. So give me one moment here, and I'll start this video.
In this video, I want to cover several topics that are all related. And on some level, they're really simple, but on a whole other level, they tend to confuse people a lot. So hopefully we can make some headway. So a good place to start, let's just imagine that I have some type of container here. Let's say that's my container. And inside of that container, I have a bunch of water molecules. It's just got a bunch of water molecules. They're all rubbing against each other. It's in its liquid form. This is liquid water. And then inside of the water molecules, I have some sugar molecules. Maybe I'll do sugar in this pink color. So I have a bunch of sugar molecules right here. I have many, many more water molecules, though. I want to make that clear. I have many, many more water molecules in this container we're dealing with. Now, in this type of situation, we call the thing that there's more of, the solvent. So in this case, there's more water molecules, and you can literally just view more as the number of molecules. Now, I'm not going to go into a whole discussion of moles and all of that because you may or may not have been exposed to that yet, but just imagine whatever there's more of, that we're going to call the solvent. So in this case, water is the solvent. And whatever there is less of, so the more, water is a solvent, and in this case, that is the sugar, that is considered the solute. This is the solute. So the sugar doesn't have to be sugar. It could be any molecule that there's less of than the water in this case. Sugar is the solute. And we say that the sugar has been dissolved into the water. Sugar has been dissolved into the water. And this whole thing right here, the combination of the water and the sugar molecules, we call a solution. We call this whole thing a solution. And a solution has a solvent and a solute. The solvent is water. That's the thing doing the dissolving. And the thing that is dissolved is the sugar. That's the solute. Now, all of this may or may not be review for you, but I'm doing it for a reason, because I want to talk about the idea of diffusion. Diffusion. And the idea is actually pretty straightforward. If I have, we'll say the same container. Let me do a slightly different container here, just to talk about diffusion. We'll go back to water and sugar, especially back to water. Let's say we have a container here. Now let's say it just has it just has some air particles in it. It could be anything, oxygen or carbon dioxide. So let me just draw a couple of air molecules here. So let's say that is a gaseous, just for the sake of argument, gaseous oxygen. So each of this is an O2, each of those, right? And let's say that this is the current configuration, that all of this is a vacuum here, and that there's some temperature. So these water molecules, they have some type of kinetic energy. They're moving in some type of random directions right there. So my question is, what is going to happen? What is going to happen in this type of container? Well, any of these guys are going to be randomly bumping into each other. They're more likely to bump into things in this down left direction than they are in the upright direction. So if this guy was happening to go in this down left direction, he's going to bump into something and then ricochet into the upright direction. But in the upright direction, there's nothing to bounce into. So in general, everything is moving in random directions, but you're more likely to be able to move in the rightward direction. When you go to the left, you're more likely to bump into something. So it's almost common sense. Over time, if you just let this system come to some type of equilibrium, and I'm not going to go into detail on what that means. You can watch the thermodynamics videos if you'd like to see that. You'll eventually see the container will look something like this. I can't guarantee it. There's some probability it would actually stay like this, but very likely that those 5 particles are going to get relatively spread out. This is diffusion. And so it's really just the spreading of particles or molecules from high concentration to low concentration areas.
Also, if I add heat, if I start heating this up, those molecules are going to diffuse a lot faster. Why do you think that would be the case?
Because they'd want to get farther away from each other because of the heat. But if it was colder, they'd get closer together.
Close. When you add heat, you're adding more kinetic energy into this. And we haven't talked about this in lecture yet. But when you're adding heat, you're adding more kinetic energy. So they have their own energy, inherent energy that causes them to bounce around. But when you add heat, you're adding even more energy to them. And that's going to allow them to diffuse out a lot faster.
Right? In this case, the molecules are going to spread in that direction from a high concentration to low concentration area. Now you're saying, Sal, what is concentration? And there's many ways to measure concentration, and you can go into molarity and molality and all of that. But the very simple idea is how much of that particle you have per unit space. So here you have a lot of those particles per unit space, and here you have very few of those particles per unit space. So this is a high concentration, and that's a low concentration. So you can imagine other experiments like this. You could imagine a solution like, let's do something like this. Let's say I have 2 containers. Let's say 2 containers. Let's go back to the solution situation. So this was a gas, but I started off with that example. So let's stay with that example. Let's say that I have a door right there that's larger than either the water or the sugar molecules. On either side, I have a bunch of water molecules. I have a bunch of water molecules on either side, just like that. On either side. So I have a lot of water molecules. So if I just had water molecules here, they're all bouncing around in random directions. And so the odds of a water molecule going this way equivalent to odds of a water molecule going that way, assuming that both sides have the same level of water molecule, otherwise the pressures would be different. But let's say that, the top of this is the same as the top of this. So there's no more pressure going in one direction or another. So, if for whatever reason, a bunch more water molecules were going in the rightward direction, then all of a sudden this would fill up with more water. And we know that isn't likely to occur. So this is, this is just a solution with, or this is just a kind of 2 containers of water. Now let's put some solute in it. Let's dissolve some solute in it. Let's say we do all the dissolving on the left-hand side. So we put some sugar molecules on the left-hand side. And these are small enough to fit through this little pipe. That's one assumption that I'm making. So what's going to happen? All of these things have some type of kinetic energy. They're all bouncing around. Well, over time, the water is going back and forth. This water molecule might go that way, that water molecule might go that way, but they net each other out. But over time, one of these big sugar molecules will be going in just the right direction to go through. Maybe this guy, instead of going in that direction, he starts off going in that direction. He goes just through this tunnel connecting the 2 containers, and he'll end up there, right? And this guy will still be bouncing around. There's some probability he goes back, but there's still more particles, more sugar particles here than there. So still there's more probability that so this guy's on that side, so let me cross him out. But there's still more probability that one of these guys will go to that side than one of these guys will go to that side. So you can imagine if you're doing this with gazillions of particles, I'm only doing it with 4, over time, the particles will have spread out so that their concentrations are roughly equal, so that maybe you'll have 2 here over time. But if you're when you're only dealing with 3 or 4 or 5 particles, there's some probability it doesn't happen. But when you're doing it with a gazillion and they're super small, it's a very, very, very high likelihood. But anyway, this whole process, we went from a container of high concentration to a container of low concentration, and the particles would have spread from the low concentration container to the high concentration container. So they diffused. This is diffusion. This is diffusion. And just so that we learn some other words that tend to be used with the idea of diffusion, when we started off, this had a higher concentration. The left-hand side container had higher concentration. Higher concentration. Higher concentration. It's all relative, right? It's all higher than this guy. Higher concentration, and this right here had a lower concentration. Lower concentration, and there are words for these things. This solution with a high concentration is called a hypertonic solution. Let me write that in yellow. Hypertonic solution. Hyper, in general, meaning having a lot of something, having too much of something. And this lower concentration is hypotonic solution, lower concentration. You might have heard maybe one of your relatives, if they haven't had a meal in a while, say, I'm hypoglycemic. That means that they're feeling lightheaded. There's not enough sugar in their bloodstream and they want to pass out, so they want a meal. If you just had a candy bar, maybe you're hyperglycemic, or maybe you're just hyper in general. But these are just good prefixes to know. But hypertonic, you have a lot of the solute, so you have high concentration, and then in hypotonic, not too much of the solute, so you have a low concentration. These are good words to know. So in general, diffusion, if there's no barriers to the diffusion like we had here, you will have the solute go from a high concentration or hypertonic solution, if they can travel, to a hypotonic solution, to a hypo where the concentration is lower. Now, let's do an interesting experiment here. We've talked about diffusion, and so far we've been talking about the diffusion of the solute, right? And in general, and this is not always the case, if you want to be as general as possible, the solute is whatever you have less of, the solvent is whatever you have more of. And the most common solvent tends to be water. But it doesn't have to be water. It could be some type of alcohol. It could be it could be mercury. It could be a whole set of molecules, but water in most biological or chemical systems tends to be the most typical solvent. It's what other things are dissolved into. But what happens if we have a tunnel where the solute is too big to travel, but water is small enough to travel? So let's think about that situation. Let's think about that situation. In order to think about it, I'm going to do something interesting. Let's say we have a container here. Let's say we have, actually I won't even draw a container. Let's just say we have an outside environment that has a bunch of water. This is the outside environment. And then you have some type of membrane. You have some type of membrane here. That's a membrane. Water can go in and out of this membrane. So it's semi-permeable. Well, it's permeable to water, but the solute cannot go through the membrane. So let's say that the solute is sugar. So we have water on the outside and also and also inside the membrane. So these are little small water molecules. This is a membrane right here. And let's say that we have some sugar molecules again. I'm just picking on sugar. It could have been anything. So we have some sugar molecules here that are just a little bit bigger, or they could be a lot bigger. Actually, they're a lot bigger than water molecules. So you have a bunch of, and I only draw 4, but you have a gazillion of them, right? But you have that much more water molecules. I'm just trying to show you have more water molecules than sugar molecules. And this membrane is semi-permeable. Semi-permeable. Permeable means that it allows things to pass. Semi-permeable means it's not completely permeable. So
semi-permeable means that certain things will be able to pass through, but others will not.
Well, in this context, I'm saying I allow water to pass through the membrane. So water can pass. Water can pass. But sugar cannot. Sugar is too large. Sugar cannot. And the reason why in this case is because the sugar molecule is too big. So if we were to zoom in on the actual membrane itself, maybe the membrane looks like this. I'm going to zoom in on this membrane. So I'm going to zoom in on this membrane. So it has little holes in the membrane, just like that. And maybe the water molecules are about that size, so they can go through those holes. So the water molecules can go back and forth through the holes, just like that. But the sugar molecules are about that big, so they cannot go through that hole. They're too big for this opening right here to go back and forth between them. Now, what do you think is going to happen in this situation? So first of all, let's use our terminology. Remember, sugar is our solute, water is our solvent, semi-permeable membrane. Which side of the membrane has a higher or lower concentration of solute?
So you guys, which side has a higher or lower concentration of the solute, the left or the right side? This right here.
Left side.
The left side having a higher or lower concentration of solute?
Higher solute. Higher concentration.
The left side, the solute meaning the sugar.
Yeah, I mean this one, it's right in the computer. It's half sugar, it's half more solute. High concentration with sugar.
Is on the left or the right side?
Left for my computer. Right to me.
This is the water. This is the water. And this is the left side.
No, the other one.
The other
right
side, the right side, correct.
Yeah.
So which side has, if the membrane is permeable to water, is the water going to diffuse from the left to the right or from the right to the left?
From left to right, according to osmosis. If it is more water in the...
Yes, so there's going to be more water over here. If you look, there's 6 water molecules here, there's 6 here. If we're talking about osmosis, and that the membrane is permeable to water, the water is going to move from the left to the right.
Left to right, yes.
When we're talking about diffusion of solute and the sugar, is the sugar going to move from the right to the left or from the left to the right?
Supposed to go from right to left, but because it is not permeable, cannot able to pass through.
Well, if the sugar can pass through right now, scratch this out right here.
Yeah, because it's high concentration can pass to the left.
It's going to move to the left, right?
Yeah.
You're correct.
Well, the inside does. The inside is hypertonic. Hypertonic. So I could write here, hypertonic. The outside has a lower concentration, so it's hypotonic. It's hypotonic. out here, lower concentration of solute. Now, if these openings were big enough, based on what we just talked about, these guys, these guys are bouncing around. The water is traveling in either direction and equal probability. Or I'm gonna talk about that in a second. If everything was wide open, it would be equal probability. But if it was wide open, these guys eventually would bounce their ways over to this side, and you would probably end up with equal concentrations eventually. And so you would have your traditional diffusion, where high concentration of solute to low concentration of solute. But in this case, these guys, they can't fit through the hole. Only water can go back and forth. If these guys were not here, water would have an equal likelihood of going in this direction as they would be going in that direction. A completely equal likelihood. But because these guys are on the right-hand side of, or in this case, on the inside of our membrane, this is our inside of our membrane zoomed up, these guys, it's less likely, because these guys might be in the kind of the approach position of the holes, it's that slightly less likely for water to be in the approach position for the holes. So it's actually more probable that water could enter than water exit. And I want to make that very clear. If these sugar molecules were not here, obviously it's equally likely for water to go in either directions. Now that these sugar molecules are there, these sugar molecules might be on the right-hand side. They might be blocking, I guess the best way to think about it is blocking the approach to the hole. They'll never be able to go through the hole themselves, and they might not even be blocking the hole, but they're going in some random direction. So if a water molecule was approaching, it's all probabilistic, and we're dealing with gazillions of molecules. It's that much more likely to be blocked to get outside. But the water molecules from the outside, there's nothing blocking them to get in, so you're going to have a flow of water inside. So in this situation, with a semi-permeable membrane, you're going to have water, you're going to have a net inward flow of water. And so this is kind of interesting. We have the solvent flowing from a hypotonic situation to a hypertonic solution. But it's only hypotonic in the solute. That's when you talk about the solute. And it's only hypertonic when you talk about the solute. But water, if you flip it the other way, if you view sugar as the solvent, then you could say we're going from a high concentration of water to a low concentration of water. I don't want to confuse you too much. This is what tends to confuse people. But just think about what's going to happen. No matter what, in what situation, the solution is going to do what it can to try to equilibrate the concentration, to make the concentrations on both sides as close as possible. And it's not just some magic. It's not like the solution knows. It's all based on probabilities and these things bumping around. But in this situation, water is more likely to flow into the container. So it's actually going to go from the hypotonic side, when we talk about low concentration of solute, to the side that has high concentrations of solute, of sugar. And actually, if this thing is stretchable, more water will keep flowing in, and this membrane will stretch out. But we won't go into too much detail here. But this idea of water, of the solvent, if in this case water is the solvent, of water as a solvent diffusing through a semi-permeable membrane, this is called osmosis. Osmosis. You've probably heard learning by osmosis. If you put a book against your head, maybe it'll just seep into your brain. Same idea. That's where the word comes from. This idea of water seeping through membranes to try to make concentrations more equal. So if you say, well, you have high concentration here, low concentration here. If there was no membrane here, these big molecules would exit. But because there's this semi-permeable membrane here, they can't. So the system, just probabilistically, no magic here, more water will enter to try to equilibrate the concentration. And eventually, if maybe there's a few molecules out here, not as high concentration here, eventually if everything was allowed to happen fully, you'll get to the point where you have just as high a concentration on this side as you have on the right-hand side, because this right-hand side is going to fill with water and also probably become a larger volume. And then once again, the probabilities of a water molecule going to the right and to the left will be the same, and you'll get to some type of equilibrium. But I want to make it very clear. Diffusion is the idea of any particle going from higher concentration and spreading into a region that has a lower concentration, kind of just spreading out. Osmosis is the diffusion of water. And usually you're talking about the diffusion of water as a solvent. And usually it's in the context of a semi-permeable membrane where the actual solute cannot travel through the membrane. Anyway, hopefully you found that useful and not completely confusing.
So here's one about osmosis. We're gonna hit this a couple times here. Next 3 videos.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules, such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of 2 solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
So tonicity always has to do with the concentration of the solute. A solution that's hypotonic will have more water in it than a solution that's hypertonic. And if those two are put together, the water from the hypotonic solution is going to move into the hypertonic solution.
What happens to a cell when it's placed in a hypotonic solution versus a hypertonic solution? A hypertonic solution is simply a solution that has a lower concentration of solutes, dissolved substances, compared to the inside environment of a cell. For example, water is considered a hypotonic solution since it is very low in solutes. A hypertonic solution is a solution that has a higher concentration of solutes compared to the cell's inside environment. For example, water with a lot of salts is considered a hypertonic solution since it is very high in solutes. How can we examine the effects of a hypertonic solution on a cell? You might be thinking, well, I'm made out of 50 to 70 trillion cells. If I drink this, I'll know what happens to cells in a hypertonic solution. But do not do that. Drinking salt water can be very dangerous as it severely dehydrates you. Nausea, vomiting, and even hallucinations are just a few
You guys, why would drinking salt water drinking seawater be toxic to you?
It sucks out more water than what you just drank.
It sucks the water out of where?
I don't know specifically where.
It sucks it out of your cells. The seawater is more constant, has more solute in it, so it's hypertonic relative to your cells. And so if you drink seawater, if you drink salt water from the sea, it's more hypertonic than what's in your cells. So your cells are hypotonic with regards to the seawater. What's going to happen to the water within your cells at that point?
Are they going to want to dilute the salt water that you're drinking?
They are. So they're all the water in your cells is going to leave to go into go into your bloodstream where all the salt water is. And when you shrink your cells, you're going to kill your cells if you're not careful. If you don't get help pretty quickly, you're going to die.
Symptoms of drinking massive amounts of salt water. Ask anyone who's been stranded at sea, which is unlikely because people who drink seawater eventually die due to kidney failure. So what you really need is
So what they say when you die of kidney failure, what you've done is you've essentially drained all the water out of your system.
Lab animal to conduct your test. Darn, she's too smart. Time to try her older brother. Well, that was a bad experiment. What's a better way to test how a cell reacts when placed in a hypertonic solution? Some eggs, like chicken eggs, are surrounded by a hard outer membrane, which protects the semi-permeable inner membrane. This membrane is semi-permeable because it allows substances to leave and some substances to enter. In particular, the semi-permeable membrane allows water to both enter and leave the cell, but large molecules, like salt or sugar, are too big to pass through. The question is, how can we examine the semi-permeable qualities of this inner membrane with the hard eggshell in the way? Dissolve the outer shell by placing an egg in vinegar. Vinegar has a chemical called acetic acid. Eggshells contain calcium carbonate. When calcium carbonate in the eggshell comes in contact with acetic acid, a chemical reaction occurs. Calcium carbonate will break down to form carbon dioxide, water, and calcium acetate, thus dissolving the calcium carbonate shell. Within seconds, you'll see bubbles form. These bubbles are carbon dioxide that are produced when acetic acid breaks down the eggshell. Within 3 days, you should have a semi-permeable membrane, free of the hard outer shell, to conduct your experiment. Before starting our experiment, we will first mass our egg using a scale. Egg A has a mass of 94 grams. Egg B has a mass of 103 grams. Egg A we will place in a hypotonic solution, water. Egg B will place in a hypertonic solution, thick and sugary corn syrup.
So if you look at what's going on with this egg that they just put in the sugar, that's would go on in your cells if you drink a bunch of seawater. Is you're just the cells are slowly gonna shrink. And remember how structure is very much related to function. If you change the structure of the cells by shrinking them, they're not gonna be as functional.
Egg A now has a mass of 97 grams. Egg A increased slightly to 103% of its original weight. We can conclude when a semi-permeable cell is in a hypotonic solution, water will enter the cell. Egg B now has a mass of 59 grams. Egg B decreased to 57% of its original weight. We can definitely conclude that when a cell is in a hypertonic solution, water will leave the cell. In fact, this is evident by the layer of water sitting on top of the corn syrup. However, 103% change in mass is not very significant. So how about we try this experiment again, but switch the eggs? This time, egg A is going in the hypertonic solution, which is still on the right. Egg B is going in the hypotonic solution, which is still on the left. After several hours, mash your eggs again. Egg A went from 97 grams to 55 grams. Egg A has decreased to 57% of its original weight. Egg B went from 59 grams to a whopping 105 grams. Egg B has increased in weight to 177%. From this experiment, we can definitely conclude that when a cell is in a hypotonic solution like water, water enters a cell. On the other hand, when a cell is in a hypertonic solution like corn syrup, or more realistically salt water, water will leave the cell. Now that you're done with this experiment, time to play with those shellless eggs. No! You're just gonna...
One last one.
Oh, when will you be back? You guys gonna be gone all day? Like 6 o'clock all day or you're usually already on?
I don't. I'm driving separate, so no, I'm gonna be back by 4-ish. Well,
Yeah. But you know that don't stop there. You almost came home and literally left with each other.
Before we take a look at how osmosis and tonicity affect a cell, let's review what each of these terms means. Osmosis represents the diffusion of water across a semi-permeable membrane. The term tonicity refers to the relative solute concentration of two environments separated by a semi-permeable membrane. In other words, by comparing the tonicity of the solutions, you can determine the direction in which osmosis will occur. Demonstrate how tonicity affects a cell. Let's place some red blood cells into a beaker containing pure water. In this case, the solute concentration is greater inside the cells than in the surrounding water. In other words, the contents of the cells are hypertonic in relation to the hypotonic contents of the beaker. Because of this, osmotic pressure results in the diffusion of water across the membrane and into the cells. Over time, if enough water enters the cells, the cell membranes may burst. This is called lysis. Now let's place the red blood cells in a beaker containing a solution of salt, such as sodium chloride. Since the contents of the beaker are hypertonic in relation to the interior of the cells, the water within the cells will diffuse across the membrane and into the contents of the beaker. This causes crenation, or shrinking of the cells. If the red blood cells are placed in a beaker whose contents match the tonicity within the cells, then there is no net gain or loss of water. The environments within the beaker and inside the cells are said to be isotonic or the same. An important thing to remember is that osmotic pressure always causes water to move from a hypotonic environment toward a hypertonic environment. In other words, water moves toward areas of high salt or sugar concentrations.
This simple process is used to drive the operation of our kidneys and explain some of the physiological consequences of diseases such as diabetes.
So drinking a bunch of salt water or sea salt water, what will that do to all your red blood cells?
It would shrink them.
What do you remember what that was called?
No, the only one I remember is the isotonic, which is where it stays the same.
Okay, it's called crenation. Spelled C is in Charlie, R is in Romeo, E is in Echo, N is in November, E is in Alpha, T is in Tango, I is in India, O is in Oscar, N is in November. Don't bite me. Sorry, just disciplining a cat here in my lap. So on a lab quiz, I could ask you to identify certain parts of the microscope. What would be the total magnification if the ocular lens were 20x and the objective lens was 100x?
200x?
Would it be 200x or would it be a little higher than that?
No, 2000.
Correct. Yeah.
Yeah, forgot a zero.
Well, that works. Image inversion. When it's exactly opposite of what you see, it's the top becomes the bottom, and the right is now the left. So when you move the slide to the right, the image in the that you're viewing is going to move to the left. Parfocal meaning you're seeing part of it. What does it mean by working distance? When is the working distance the greatest? Let's put this differently. When is the field of view under the microscope the greatest? When is it the least?
The field of view is the greatest when you have the smallest objective, which is the red one, and then it's the least when you have the bigger objective.
Right, because the more you magnify it, the more you bump up your magnification, the lower your field of view is going to be. Why must an image be centered before increasing the magnification? Because that's where your biggest field of view is at the least amount of magnification. As you increase it, your field of view goes down, so if it's not centered, you may not be able to see it anymore. This was one of the terms from that slide that that talked about the different body parts. Buccal means cheek. Identify the different parts of the cell. You're going to want to use your textbook for this one. Maintaining steady state of a cell, meaning you're maintaining the homeostasis of the cell. So it allows nutrients to enter the cell. It's selectively permeable to allow nutrients to enter the cell, but keeps out other undesirable substances. So passive meaning moving down the gradient. The differences, the gradients drive the movement. Active meaning you're providing some form of energy to power the transport process, and it's against the gradient. And remember, a gradient is the difference between one side versus the other side, and it's in how much of whatever you're looking at is there. So if you're looking at sodium ions, sodium positive charges, how many sodium positive charges are on one side? How many are on the other side? If you're looking at a gas gradient, a pressure gradient, if the pressure is higher on one side than it is on the other, if the pressure is higher on the left side than it is on the right side, the pressure is going to move from a high to a low every time, unless you put some form of energy into it to block it from being able to move down the gradient and forcing it to move up the gradient. In this experiment, this uses dialysis tubing, and you put a concentrated, bear with me just for a second here, you put a concentrated starch solution in the dialysis tubing. You drop some iodine into the bottom of the beaker, and wait. And what is learned by this is glucose does not interact with iodine, but starch does. So glucose is able to move out of the dialysis tubing. The starch is not. And that's why when you put iodine in the water in here, you see that when the iodine diffuses into the bag, it darkens up the starch. But there's no starch that actually diffuses out into the bag. So the iodine molecules are small enough to pass freely through the membrane. However, the starch molecules are complex and too large to pass through. Thus, the iodine diffused into the tube with the starch. But the starch never left the tubing. Osmosis, all organisms must maintain an optimal internal osmotic environment. Osmosis is that passive movement of water down its concentration gradient. Free water molecules are ones that are not highly associated with solute. So if we look at this beaker right down here, the free water molecules are ones like this one and this one up here. This is not a free water molecule because it's working to keep this green solute dissolved. Like experiment. How many of you work currently in the healthcare field?
I do.
When you, have you ever, have you ever set up an IV? Or seen one set up?
Yes.
Okay. What's the concentration of the saline solution that they tend to put in an IV bag?
Is it
0.9?
It's 0.9. Why is it 0.9% sodium chloride?
It looks like isotonic is not more concentrated. It's not affecting the cell.
Yeah, it's in there because it's the same tonicity salt-wise as the rest of your blood.
Body cell.
Say it again?
Cell of the body.
Yeah, so it's the same tonicity as the rest of the cells of your body. And so it's not going to cause your cells to either get lysed or undergo crenation and shrink. So tonicity refers to the relative concentrations of a solute inside and outside of the cell. For the last part of our lab today, we have these worksheets. And I personally, I'm not sure if you'll be able to finish. We have about, how much time do we have? We have about 40 minutes. You can print, I personally would type them out and you're going to submit these to me. So you're going to email these to me. Actually, you know what? I'll probably put a turn it in option. I'll develop a turn it in option on the Dropbox here and have you submit them there. Does it actually give you a microscope there? So you're going to label the microscope. You've got a word bank down here. What is the total magnification of the typical microscope? The scanning objective, it's about 4x. Oh, the total magnification, sorry. Ocular lens is about 10x. With the scanning objective, it's about 40x. Because the scanning objective is 4. I think it's this one right here. And the ocular lens is 10. What part of the microscope should you never touch when observing a specimen on low power, high power, or oil immersion? I would say that's probably the objective lens. And the reason is so that you don't damage it or break it or smudge it. If you smudge it, it's going to be a lot harder to see through it. Parfocal meaning that one eye sees its field of view, the other eye sees its field of view. That wasn't what I wanted. Define diffusion. What is osmosis? Is it an active or passive process? Briefly explain what happens to red blood cells when it's placed into an isotonic situation, a hypertonic solution, or a hypotonic solution. What effect does increasing the temperature have on the rate of diffusion? If you remember, I talked about during that Khan Academy video, and I said what would happen if you introduced some heat into this? How would that change the rate of how fast the diffusion occurs?
Yes.
So you want these done by next class?
What I would do is I would, let's say, have them turned in by midnight the night before class. So next Monday night at midnight. And I will make that Dropbox here within the next couple hours.
I just want to make sure I'm on the same page. You're talking about the worksheets for Lab 2?
Yes.
Awesome.
Before Monday or before the lab?
Before, by midnight next Monday.
Before the lecture, okay.
Yeah, before the so we have lab on.
When?
So sorry. Let me do this differently. Let me say this differently. Today is Wednesday. So before midnight on Tuesday night.
Okay.
So I just want it done before we have lab on Wednesday morning.
Next Wednesday, okay.
Yes.
Thank you.
We're not having class on Monday, are we, because of
No,
Labor Day?
We will not. We won't. But we will have class on Wednesday.
Okay.
And it's only those two worksheets, right? It's not the third one that's below that. That's more of a study guide.
Well, I think that third one is also one of the worksheets. That third one.
So all three of them.
Yes.
Okay.
What else was I going to say? Give me one moment here. If you want to, I put up the PowerPoint for Chapter 2. I've also put up the review worksheet for Chapter 2. If you feel so inclined, you can start looking at that too. And I will get give me a day or two to get up the Zoom videos, and I will get them up. I'll get up every recorded video I have for our class. Um, I honestly am going to learn here really quickly how we do Zoom rooms, breakout rooms on Zoom. Give me one moment here. Maybe. So I'm just going to assign you into, there's 19 participants here. I'm going to sign you up into 4 different rooms. Or would you prefer to work in smaller groups than that?
That's good.
That should be fine.
Hey, do me a favor, if you, I would like you to stay. If you think you know all the answers to those questions, help the other people in your group. And I need you, please stay for at least 15 minutes and let me write down your names because I have to turn in an attendance sheet for your financial aid and so forth with the university, with the college. So stay in there just long enough for me to at least get the roll done, okay? And get attendance taken. Will Spar, are you there? Will Spar, can you unmute your microphone?
Did today.
How is it going?
Good. Yeah, you remember was it on Monday? This is you told me you're gonna help me with the connect.
Yes, can we do it after class? Is
Yeah,
that
sure.
okay? Were you able to call them?
I was able to call them, but apparently they're not able to really help me. So
Okay. You're probably not the only one. I think I got an email or a text from someone else in the class saying that they weren't able to get what they needed. Yes, I can do that.
all right, thank you. And I didn't know that there was lab one assignment due today.
What was the lab one assignment that was due today?
Somebody told me lab one assignment is due today, so I don't know.
Let me look. Give me 1 second here.
Was it an assignment or worksheet?
I think it was probably an activity. But I'm going to actually I don't think there was anything due today. Give me 1 moment here.
I think it's not due until September 16th for the Lab 1 simulation.
Yeah.
Yeah.
Yeah, it's not. If you look here, do you see my screen?
Hold on, I'm gonna go back. Yeah.
It tells you when the due dates are.
Okay. Okay.
Yes, that lab due date for the virtual lab is the 16th.
Is the 16th. The lab one is okay.
Yes.
All right. But then I have to have access to connect before I can do those, right?
Yes, you have a 2-week free period with it. Let me.
I just want to be able to get access to connect, and then I can just look at them and then do it.
Yes, I understand that. When we are done with class at 10:45, let's give them a call.
Okay. Okay.
I'm gonna call them and you and I are, and I think there's one other student. We're gonna get on Zoom. We'll stay on Zoom and I will call them and we'll make sure that you get what you need, okay?
Okay. Thank you. Appreciate it.
You're welcome. How are the worksheets coming along?
Well, we just got on there.
I know, I understand. I'm just joining to ask.
Yeah. I'm just gonna print everything and just do it, and then just take a picture and send it to you at the Dropbox.
I'm gonna have you put it in the Dropbox.
Yeah, that's what I'm saying. I'm gonna just I could just scan it. I'll scan it and just put it in the Dropbox.
Okay. I need to be able to read it. That's my major concern, is just make sure that I can read what you read it.
Sure.
How are the rest of you doing?
Okay.
I'm doing fine.
Is there anything I can help with? Any questions?
For the lab, the first worksheet where it says to label all the parts of the microscope, do we have to write the function down on that sheet also?
Don't think so. Let me come back and look at it really quickly here.
There's still gonna be space for that.
Yeah, I think that first part is just labeling it.
Okay.
Buggy.
So yeah, that first one is just labeling it.
Okay.
Okay, I'm gonna go and join one of the other groups. If you have questions for me, you still feel free to text me or email me. I mean, usually texting is a lot easier for me to get back to.
Okay.
Are we done then?
Yeah, we can be done here. I got what I needed from roll, so yes, we can be done here.
Okay, thank you.
Okay. Do I have to?
Stay, if you want to, and I know your name isn't 80. What is it? What's your name?
Right. Just forget about the middle name. I'm James. James Lonka.
James. Okay. I will try to remember that because when it comes up, that is what it comes up as. But I'll try to remember that.
Yeah, I'll change it. I don't know why it says my middle name there.
James, if you want to, just come back at 10:45.
10:45. Okay. Okay. So you want me just join the class again at 10:45?
Yes.
Okay. 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 So we can be done here right
Yeah.
now? Okay.
Yes. How is it going?
It's going well. We're just all working independently and then asking each other questions as we need to.
Can I help you at all? Any questions I can answer?
Not so far. I'll go in the main room if we can't figure it out.
Okay. All
Thank
right.
you.
You're welcome. How is it going?
Good. Good.
It makes it easier to see.
It does make it easier to see. I see what you guys are doing. One thing that you can say with that question is that it makes it easier to see because the specimens tend to be very clear. And so adding a little bit of color to them allows you to see them better. The specimens tend to be, if you saw him pull off that onion skin, the epithelial tissue of the onion, they don't have, doesn't have any color to it. And so to be able to see the different structures, the color helps you to see those structures.
Oh, like that?
Yes. Or you know, the other thing that you can put in there is the clear, I would say it makes the clear specimen easier to see. Yeah, or the transparent, either one. Transparent or clear.
All right. Okay. And then we just put the numbers like on the side. Is that all right?
Yep, that works.
Okay. Okay, so this is the other worksheet. Is there not a word bank for this one?
Maybe not.
Okay.
It will be if you have your books or access to your books. It's all in there.
I think number one's mitochondria.
What'd you say?
Mitochondria.
Mm-hmm.
I think two is a lysosome. Three is Golgi.
Wow.
Three, yeah, the Golgi apparatus, correct. I think number two is yeah, you're probably right. That's probably a lysosome.
Not sure what four is though.
For you know, I'm going to tell you that I think four is a lysosome. I don't have an answer key here.
Right.
Four is a lysosome. And that little number three is a vacuole.
Number two?
Number, I'm sorry, number two is a vacuole. And the reason I'm saying that is if you look right here, you guys see my mouse? I know you're sharing your screen. Do you see my mouse here right above the number four?
Right here?
The little clear thing that's on the the plasma membrane there that's putting stuff out. That looks like number two, but it's fused with this membrane.
That's like a secretory vessel.
Yep, or vacuole. They're also called vacuoles.
How do you spell vacuole?
You have most of it there. V is in Victor, A is in Alpha, C is in Charlie, U is in Uniform, O is in Oscar, L is in Lima, E is in Echo. Yeah.
And that's for two and the one over here, even though that's not labeled?
Yes.
Okay.
Yes.
And 5.
Number 10 is the same thing as that what I was just trying to describe there for number 4. What did you say it was?
I said a secretory vessel.
Yep.
Okay.
I'm going to go and enter one of the other labs.
All right.
One of the other groups.
I think.
Fine.
It's good.
Good. Can I help you with anything?
What?
Can I help you with anything?
We're doing all 3 of those worksheets, correct?
Yes.
Okay.
Kyle, so you don't have Microsoft Word? Do you, do you, do you have it on another computer? And you can chat your message. I don't mind.
tried to copy paste the worksheet in my Google Docs, but the pictures aren't like being copy paste.
I'm sorry.
Only the questions.
Say that one more time.
I tried to copy paste the worksheet in my Google Docs, but it doesn't let me copy paste the pictures, the actual pictures. Only the questions only.
Can you save them? Can you save it as a PDF?
I'm not sure. And I also don't have access to the Connect. How do I do it?
If you stay on here, I'm going to call them. You're not the only student here who's not got access to that. I'm going to call their helpline right now, and we will figure out how to get you the Connect. Give me one moment, okay? I'm going to leave this break room. Can you join back in the main room?
Yes, sir.
Okay. Okay. James?
Yes.
Give me what I'm gonna call them right now, so bear with me just for a second here.
Okay.
Professor Gessel's class is over. Do we just leave now?
Yes, you can just leave now.
All right, thank you.
You're welcome. I'm gonna put them on speaker here.
McGraw-Hill. To ensure we are providing exceptional service, this call may be recorded. Due to increased enrollment during COVID-19, we are experiencing long hold times. For many options, you can self-service on our website by visiting www.mheducation.com/customer service. That's www.mheducation.com/customer service. If you are a Barnes & Noble or Follett bookstore employee, press 1. If you are a student, press 2. All others, press 3. Please select from one of the following 5 options. To place an order, to pay for an order you already placed, or to check price and product availability, press 1. To request a copy of an invoice, press 2. If you are having issues with your access code not working, unable to view course materials, have missing assignments, or other issues with an ebook or online product, press 3. To check the status of an existing order, press 4. For all other inquiries, press 5. For K-12 school products, press 1. For college students and higher education support, press 2. We are experiencing high call volume and you may experience longer wait times. Your call is important to us and representatives will assist you as soon as possible.
Welcome to McGraw-Hill Technical Support. For quality control and training purposes, your call may be monitored. Please listen carefully as our menu options have changed. If you are an instructor needing assistance, please press 1. If you are a student needing assistance, please press 2. Your entry is invalid. Please try again. If you are an instructor needing assistance, please press 1. If you are a student needing assistance, please press 2. Your call cannot be completed at this time. Please try your call again later.
Oh, really? All right, let me try one more time.
Thank you for calling McGraw-Hill. To ensure we are providing exceptional service, this call may be recorded. Due to increased enrollment during COVID-19, we are experiencing long hold times. For many options, you can self-service on our website by visiting www.mheducation.com/customer service. That's www.mheducation.com/customer